Introduction to Diagnostic Parasitology and Clinically Relevant Cestodes. Parasites are an important cause of human morbidity and mortality in many parts of the world. Parasites are often associated with gastrointestinal infection or, in the case of malaria, a blood infection. Other human organs and systems may also be infected. Important and significant information when a patient presents with an infection that may be caused by a parasite would be patient's symptoms and clinical history, including travel history. The specimens commonly found at the parasitology bench: stool is the number one most common specimen, blood, urine and vaginal specimens, sputum, CSF, specialized specimens such as skin snips, arthropods. Arthropods are insects like ticks. Those can show up at the parasitology bench. Duodenal aspirate, and eye or corneal scraping. So first talking about stool specimens, collection, handling, and transport. Stool specimens, fresh stool in a wide mouth container. You want to examine it within 30 to 60 minutes of it being deposited into the container. We never want to scoop it out of a container that's been contaminated with urine. We want a fresh stool sample with no other contamination. 3 specimens, 2 on successive days and 1 on a different day. So you would do 2 in a row on 2 different days and then 1 on an unrelated day out of the sequence, but still within a 10-day period. Unacceptable specimens would be samples with water or urine contamination, barium contamination. So some GI tests include or involve a patient swallowing barium, so we want to avoid that. You have to wait 7 to 10 days later to collect the stool specimens. Stool samples should either be examined or preserved immediately. So the time frame after sample passage: liquid stools would be 30 minutes, soft stools 1 hour, formed stools 24 hours, and you can refrigerate those if necessary. Preservatives that are commonly used would be 10% formalin. That preserves ova, cysts, and larvae for concentration and wet mount examination. And polyvinyl alcohol, or PVA, preserves cysts and trophozoites for permanent stains. Gross examination of the stool. Consistency, you want to determine if it's formed, soft, loose, or watery. This is important. So when you look at the microscopic evidence, the type of stool it came from is going to provide some important differentiating factors, especially if it's a protozoan parasite. Trophozoites are more likely to be found in watery and mushy stools. If we talk about kind of going back to what trophozoites are in the first place, because we haven't really defined that yet, when we talk about parasites, our 2 main categories are going to be the helminths, or the worms, and the protozoans, or the single-celled organisms that are animal-like and that eat other organisms in order to survive. So those are our 2 main categories. Now the worms, we can look at eggs, also known as ova. We can look at the chunks of the worms or the whole worms themselves, and we can look at the mouth parts of the worms, so the worm in general or the egg that the worm produces. But on the protozoan side of things, and those single-celled organism sides of things, there's typically 2 main life cycle stages, and that would be the cyst or the trophozoite. The trophozoite would be the living, breathing, moving, kind of vegetative organism that would be found inside of the body, while the cyst is usually the dormant form of the organism that is most commonly ingested from the environment. So once the organism passes through the lower part of the digestive tract, all of the water is sucked out of that stool specimen, and cysts will form at that point. And then when a person defecates, usually the cysts are coming out. Unless it's super watery stool, then the trophozoite form would also pass out. If it sits in a container for a period of time, let's say like over an hour, then all those trophozoites are going to start to convert to cysts because it's no longer going to be at the appropriate temperature, which would be body temperature. It's out in room temperature, typically in a container. So we do want to examine those specimens pretty quickly. A lot of times, trophozoites are much easier to distinguish than cysts. So cysts are all like round balls. And you can measure the cysts, and there's some varying characteristics that you can look at under a microscope. But it is definitely easier to identify the trophozoite stage. So if we do get lucky enough to get some trophozoites in a sample, we can usually come up with a pretty definitive diagnosis and follow that up with our molecular or immunological testing. So now that you know a little bit about the types of parasites we're seeing, this is why the trophozoites are more likely to be found in watery and mushy stools. Because in watery and mushy stools, the moisture hasn't been absorbed from them, so you're still going to see that living, breathing, moving vegetative state. Cysts are more likely to be found in formed stools because the water has been removed, and that's that dormant form of the organism that's then going to be deposited in the stool and transferred to the next individual, either through contaminated food or water. Helminths may be found in stool samples of all consistencies. So the worms doesn't really matter. Color, brown, black, red, etc. The relationship of stool consistency and protozoan stage is depicted here. Again, I talked about the protozoans and the cysts and trophozoites. So as the stool becomes more formed, you're going to see more of the cyst stage and less of the trophozoite stage. Examine the surface for proglottids, for tapeworms, and adult worms if they're nematodes. Break up the specimen with an applicator stick to look for adult worms. Report the presence of mucus and or blood when found. And then there's 3 types of preparations for liquid and soft stool. You can do direct wet mounts, concentrates, and permanent stained smears. Prior to using the slide, you have to clean it, so render the slide grease-free by cleaning the slide with 70% ethanol. Now the main thing with stool is everything looks like a parasite. So if you just take a fecal sample from any healthy, normal individual and you look at it under the microscope, you're going to find a lot of roughage in there, they call it, a lot of material that looks a lot like eggs. So it really does take a trained eye to be able to differentiate that, especially if that individual is eating a lot of vegetables. So if they have a lot of like leafy greens and stuff, a lot of those plant organisms are going to come across in the urine as if they were like pieces of worm or the eggs of the worms. So it takes a lot of practice and skill and technique to be able to look through a stool specimen and accurately identify certain types of worms. So it's important to clean the slide so that you decrease the likelihood that there's some background nonsense that's going to mess up your view of the slide. So wet mount preparation is a rapid technique. You can visualize trophozoite motility and other parasitic stages may be seen. You place a small amount of saline on the slide, mix a small amount of stool into the saline on the slide. The stool amount should be small such that when it's mixed, it's thin enough to read through. So here you can see a glass slide, a cover slip on top of the glass slide, and you can see the consistency of the stool that's been mixed in the saline on the slide. You can read through it. Why does it have to be so thin? Because light has to be able to pass through the specimen. If we can't clearly see through the specimen, we won't be able to see the intricate details of the egg. Or if we do get lucky enough to find chunks of the worm, we won't be able to see the outlines of those chunks and the details that help us differentiate it. Carefully lay a cover slip on top to avoid the formation of air bubbles. Formalin or PVA samples can be used, although trophozoites are killed by preservatives. So if you want to look at motile organisms, you don't add those preservatives. If you don't care if they're swimming or not, then you can add the preservatives. You always want to angle the cover slip, and then when you drop it, the liquid by capillary action will kind of just diffuse out in this direction so that it reduces the likelihood of air bubbles forming. You want to examine the entire prep using the 10x objective and then examine several fields at 40x. A negative result must be confirmed by concentration techniques. So you're going to start at one end and go up, down, up, down, up, down, and then you start at the top or the bottom and go back, forth, back, forth, back, forth, looking through the entire slide. Concentration techniques. So sometimes there's not a lot of parasitic eggs or parasitic evidence, cyst trophozoites found in a stool specimen. So in order to kind of concentrate the fecal specimen down to its parasitic essence. We go through sometimes a concentration technique. So through the sedimentation process, solutions with specific gravity lower than parasitic organisms is added. We're going to mix it up into the fecal specimen. Then we use the centrifugation to pellet the sediment. Organisms are concentrated in the sediment. And then we just take a scoop of the sediment out and look at that under the microscope, and that's where the majority of our eggs should be. The formalin ethyl acetate method is the most common. So stool is mixed with formalin. Suspended stools are stained to remove large strained, to remove large particles. They're usually strained with just some gauze. Centrifugation is used to create a sediment. Sediment is resuspended in formalin or saline and mixed with ethyl acetate. The mixture is shaken vigorously and centrifuged, and 4 layers are going to form: the ethyl acetate layer, fecal debris, formalin or saline, and then the sediment containing the parasites. You do a pour-off method, which is kind of an acquired technique where you very carefully and directly pour off all of the top liquid portion while not disturbing the sediment, because the sediment is where all of our parasites are, and we don't want those being poured off. Wet mounts with and without iodine are made from sediment. If you're ever asked what type of stain is commonly used to visualize eggs or parasites in stool samples, iodine is usually our go-to stain that we use. So, for certain types of protozoan infections, we have modified acid-fast stains, which helps us detect oocysts. Stain magenta against a blue background. So here you can see this one looks like it's Cryptosporidium. Iodine stains glycogen and destroys trophozoite motility. So if you do stain with iodine, it's going to stop them from moving, but it stains glycogen, making the internal structures a little bit more obvious. Permanently stained spheres. So it's recommended that you make a permanently stained smear of each one of the 3 fecal collections that happen, just so you can always go back and reference that in case something ends up being positive later on and a tech missed it. Iron hematoxylin stain, high definition of parasitic structures, but it's a difficult procedure and it has some variability. The trichrome stain, cytoplasm of cysts and trophozoites, that is, are blue-green tinged with purple. Nuclear chromatin ingested red blood cells appear red or red-purple, and the background is green, and that one is in wide use. Blood specimens. So extra erythrocytic forms, in other words, the blood parasites that are found outside of the blood cells, can be detected in plasma or visualized in a blood wet mount. Trypanosomes and microfilaria are the ones that are going to be extra erythrocytic. We are going to learn about all these different types of trypanosomes and microfilaria, malaria, all that kind of stuff as we go through the next few weeks of content. Malaria and babesia, thin and thick smears are used. So those are going to be inside of the red blood cells predominantly. So we're going to be able to see the organisms inside. They're stained with Giemsa or Wright stain. Collect samples at about 6-hour intervals over 36 to 48 hours. Why do we want to collect at intervals? I'm going to teach you about the periodicity of certain types of specimens. Depending on where these organisms are in their life cycle of infection, some organisms may or may not be actively present in the blood or in the blood cells at the time that you do the blood collection. So it's important to collect multiple specimens to try to capture them when they are in the blood, because we don't really know when they're going to leave the liver or leave their primary organ and go into the bloodstream. Collection and preparation. Anticoagulated blood is used for most specimens. EDTA is common. Time of collection is important for certain parasites. So thin smear versus thick smear preparation. In a thin smear preparation, you can see the blood is very thin. Thin smears allow us to look for individual organisms within a specimen. Thick smear You just put a big drop of concentrated blood on there. Thick smears allow us to scan for the potential for parasites. So if we want to know a quick answer of yes or no, they probably have some parasites inside their blood, we would prepare a thick smear. If the thick smear is positive, then we would go the next step further and prepare a thin smear in order to identify that organism down to the species level in some cases. Now, this type of identification is tricky. It takes a lot of skill to be able to do this, and usually our standard 410, 40, 100 objective microscopes don't cut it. A lot of times we would need like a 200x objective to be able to properly visualize and distinguish the type of organism. So in general, in order to prepare a thin smear, you're going to place a drop of blood near one end of a slide. Then you hold another slide at a 45-degree angle and draw it into the drop of the blood. So basically here is that drop, and you would put another slide kind of at an angle here, and you would butt it up against that drop, and then you would quickly smear it to the right. So that would spread that blood all the way over to the right side. It's going to be most concentrated here, eventually fanning out to less and less concentrated. The area that you want to try to visualize is this kind of central area, and then you can always go to the left or right as needed to try to see different structures of the different parasites found. For the thick smear preparation, you touch a slide to a drop of blood, rotate the slide to form a circular film, and look at it under the microscope. So this is what you can see. In a thick smear, you can see that there's something there that shouldn't be. In a thin smear, you can blow it up and actually see like the individual parasites and be able to identify these to the species level. Since there's more than one of these inside of this cell, this looks like it's Plasmodium falciparum, which is one of the leading causes of malaria. Urine and vaginal specimens. So in urine, we're looking for schistosomiasis, centrifugation to sediment, and then you can see the eggs. We're going to learn about schistosomiasis and how it's spread through snails and how it can cause bladder cancer. Vaginal and urethral discharge. So T. vaginalis is the most common cause of STDs worldwide. This one can be added to pouch medium and incubated, and then you'll see these guys, and this is what they look like, and then they have little flagella, and they kind of spin around and swim around and all kinds of crazy stuff. So really cool if you can see some live organisms under the microscope, but very distinguishable if they're actually swimming. Sputum specimens. Common isolates would be Ascaris and Strongyloides larvae and Paragonomus westermanii eggs. And that's the larvae and that's the egg. CSF or cerebral spinal fluid specimens. Common parasites would be Acanthamoeba, Balamuthia, Echinococcus, Naegleria fowleri, Tinea solium, Toxoplasma gondii, or Trypanosoma species. This is a picture of Naegleria fowleri. We'll learn about those as we go through these slides, or the next couple weeks of slides. Specialized specimens, skin snips. So with skin snips, sometimes parasites like the Onchocerca volvulus microfilaria. These are tiny little worms that can form in a raised bump inside of the skin. You can tell that they're there because you're going to have a lack of pigmentation in that area, and you can pull up part of the skin, cut a little chunk of the skin out, and look at it under the microscope and be able to see all of these little microfilaria to diagnose that as the causative agent. Arthropods, as I mentioned, insects. The insect is usually a tick for identification because certain types of ticks are more likely to transmit Borrelia burgdorferi, which is the causative agent of Lyme disease. There's also other types of ticks that can transmit other types of infections like Rocky Mountain spotted fever and all sorts of crazy stuff. So ticks will come to the parasitology bench, and then we will look at them under the microscope to try to identify them. Now, these are really pretty ticks. You can see all their structures very clearly. When we receive ticks, sometimes they're engorged, which makes them a little bit trickier to identify. You want to provide these in a leak-proof container with 70% ethanol. The ethanol will kill the organism, but if you want to culture the mouth parts, if you want to actually try to culture the bacteria that's growing in their mouth, 70% ethanol would kill that bacteria. So you have to kind of consider, are you looking for just a tick identification, or do you want to go through with diagnosing the type of bacteria that it may be transmitting through its saliva? These have an indefinite transport time if you are just looking for insect identification. Duodenal aspirates. So duodenum and jejunum aspirates, saline wet mount, usually testing for Giardia lamblia or Strongyloides stercoralis. The string test, you use a nylon string with a weighted capsule to enter the duodenum. Then you remove it 4 to 6 hours later and do a wet mount of the mucus that's obtained. Common parasites, Clinorchus sinensis, Cryptosporidium parvum, Giardia lamblia, Isospora bellii, or Strongyloides species. This one's super gross. Eye scraping. So here we've got Acanthamoeba, Naegleria, also in contact lenses, and all open solutions. This guy in the eyeball is most likely the Microfilaria loa loa, which we'll learn about as we go through these next few weeks. Acanthamoeba is a pretty common cause of like this cloudy white patch that forms in the eye and has been associated with contact lenses. The periodicity of specimen collection. So as I mentioned, certain types of parasites, especially parasites that we're trying to isolate from the blood, will retreat into particular parts of the body, and then they'll come out into the bloodstream during certain times of the day. And we don't really know what organism is causing the infection, so we have to collect blood specimens at various times to try to catch it when it's actually in the blood. So here you can see different microfilaria and when you would most commonly see them in the blood based on their time of infection, and you can see it changes, so it's really difficult to predict when the organism is going to be present. Cellophane tape test for pinworm. Enterobius vermicularis is the pinworm. The female worms migrate from the anus at night to lay eggs in the perineal region. So with Enterobius vermicularis, we'll learn more about this worm as we go through here. But you got a male and a female that usually infect the lower digestive tract. The female will poke her little head out at nighttime while the person's sleeping and deposit eggs around the rectum. So then you would collect the eggs by putting a piece of cellophane tape over the perineal area. And then you would just peel the tape off in the morning and then look at it under the microscope. After it's been taken, the sticky side of the tape is placed on a microscope slide and scanned at low and high power magnification for the characteristically shaped eggs. Adaptations of this procedure using paddles with a sticky surface are commercially available. So sometimes these parasites are really hard to detect just by looking for them in specimens. So sometimes we have to do immunological tests or molecular tests. And molecular tests are becoming much more common. It's pretty rare that we would actually scan through a stool specimen anymore looking for certain types of parasites. There are some specialized labs, especially in the southern United States, where these types of infections are a bit more common due to the temperature and climate and all that kind of stuff. But in your everyday run-of-the-mill typical clinical lab, you're going to predominantly depend on molecular testing for this identification. But when we talk about immunologic diagnosis, parasites that invade tissue like Entamoeba histolytica, Trypanosoma cruzi, or Toxoplasma gondii are the primary organisms that stimulate antibody production. Serological tests are sometimes useful if invasive methods cannot be used for identification. So sometimes these guys embed themselves deep into tissues, and it's a really invasive procedure to be able to grab them. So we can look at the blood for the presence of antibodies against them. In most cases, however, tests for antibodies serve only as epidemiological markers, especially in endemic areas. They estimate like 90% of cat owners already have Toxoplasma gondii inside of them. So, if all of a sudden someone tests positive for those antibodies, it's like, well, do they have an active infection and is it actually going to be a problem, or were these antibodies that kind of stayed around from a previous infection? Enzyme immunoassays, EIAs, provide information about current infection in contrast with antibody tests. Several EIAs are available to detect the presence of Giardia duodenalis, Cryptosporidium, Entamoeba histolytica, and Entamoeba dispar. Antigen detection kits have replaced microscopic examination in some hospital laboratories. A growing area EIA application testing is the diagnosis of malaria. These tests are based on the principle of immunochromatographic antigen capture and use whole blood to detect malaria protein. The patient's blood is reacted with monoclonal antibody labeled with dye or gold particles. Some tests are relatively non-species specific and detect a protein, such as parasite lactate dehydrogenase or aldolase, that's common for all human Plasmodium species. Some tests detect both types of protein and provide a more complete picture of the infective agent. Treatment for a lot of parasitic infections, like if you have a tapeworm infection, you take an anti-tapeworm drug, and it wipes out any type of tapeworm you have. So diagnosis down to like the species level oftentimes isn't necessary. Now, it's cool to know like what organism caused this, and in immunocompromised individuals, it's really important to know what the causative agent actually was and how this person can prevent themselves from getting infected again. But if we're talking about like treatment's going to be the same for most tapeworm infections. Treatment's also going to be the same for most roundworm infections. And a lot of those drugs are based around the premise of interrupting their replication. So it kind of stops them from being able to reproduce or produce eggs, and then eventually they get flushed out of the body. It prevents attachment of them to your intestinal lining. So that type of treatment method, because it's so generalized, a lot of times we don't need to get it down to the species level. But malaria is a whole different, like the Plasmodium species, that's a whole complicated topic. So we'll talk a lot more in detail about anti-malarial drugs and the different types of malaria and where you'd find those different types and all that kind of stuff once we get to that lecture. Fluorescent antibody techniques. Direct fluorescent antibody techniques use monoclonal antibodies that have been developed to detect Cryptosporidium oocysts in fecal specimens. Fecal material is placed and spread on a slide. The reagent containing the antibody is added. Specimen is examined under a fluorescent microscope for a characteristic apple green structure. The DFA combination reagent for G. duodenalis and Cryptosporidium is also available. Such procedures are useful in screening large numbers of specimens during epidemiological studies. DFAs have greater sensitivity and specificity compared with routine stains. The quantitative Buffy coat procedure uses the fluorescent dye acridine orange to stain nuclear material for detecting malaria organisms in blood. A microhemocrit tube is coated with fluorescent dye. After centrifugation, the parasites can be viewed in a small area at the top of the erythrocyte column. A thin smear still must be made for definitive identification. And then those molecular methods. So molecular methods for parasite identification are rapidly being adopted by clinical labs. Methods can include classic and real-time polymerase chain reaction or PCR assays. Molecular methods of detection exist that identify several organisms, including malaria parasites. There's multiplexed PCR panels that are commercially available for GI pathogens that can detect a significant number of viruses and bacteria and a limited number of parasites, like Giardia duodenalis, Entamoeba histolytica, Cryptosporidium, and in some cases, Cyclospora. So it's one PCR with multiple primers, and those primers are designed to identify many different species from one tube, and those are becoming increasingly popular. Dientamoeba fragilis, blastocystis, and microsporidia do not yet have a rapid identification procedure. So medically important parasitic agents. So I talked previously about the two major categories. We have helminths and protozoa. Helminths are the worms. And three kind of subcategories in here are cestodes, trematodes, and nematodes. Cestodes are your tapeworms, trematodes are your flukes, and nematodes are roundworms. For protozoa, we have Lobosea, also known as amoeba, Trypanosoma, Mastigophora, or the flagellates, Leishmania, the ciliated ones, which really there's only one ciliated one, and the sporozoa, also known as apicomplexans. So protozoa are mostly named based on how they move, while helminths are mostly named based on the shape of the adult worm. And then this is a picture showing eggs of various helminths. So helminth infections in animals are caused by flukes, tapeworms, or roundworms. Modes of transmission: directly ingesting eggs, or by ingesting larvae in an intermediate host, or through direct larval penetration of the skin. Adult worms do not multiply in the human body. The number of worms is related to the number of eggs or larvae that the person ingested. The pathological consequences and severity of infection are related to the number of adults present, commonly referred to as the worm burden. With tapeworms are a little different. Usually people only get infected with one tapeworm. So even if they consume 500 eggs, only one of them will actually grow into a full adult worm. Patients with only a few adult worms are usually asymptomatic, whereas patients with a large number of adults show clinical symptoms. Most parasites inhabit the intestinal tract, but species also infect the liver, lungs, lymphatics, and blood vessels. Tapeworms. So tapeworms or cestodes, these are the main guys, the main players that we are going to talk about in this lecture. So Diphyllobothrium latum is the fish tapeworm. Tinea saginata is the beef tapeworm. Tinea solium is the pork tapeworm. Hymenolepis nana is the dwarf tapeworm. Hymenolepis diminuta is the rat tapeworm. And Dipylidium caninum is the dog tapeworm. Now with Dipylidium caninum, we aren't going to go into the details or specifics of that one. You won't have to be able to identify that one, but knowing it's the dog tapeworm is helpful. Eventually you may encounter this in clinical specimens if you work in a veterinary field. Very rarely this can infect children, but we're not really going to get into all the details of this one. So cestodes, the platyhelminths, all require an intermediate host. An intermediate host is part of the life cycle, and we're going to talk about the life cycle of several of these worms. Adult morphology is from 3 millimeters to 10 meters in length. 10 meters, that's not a typo, so massive. They're ribbon-like with a head like scolex and suckers for attachment to the host. They are segmented, and each one of the segments are called proglottids. They're hermaphroditic, in other words, self-fertilizing. So here's the head with the scolex, or the head or scolex, and the scolex allows it to attach to typically the intestine. And then you have all of these individual segments called proglottids. Egg morphology. Most eggs in this group contain a tapeworm embryo with 3 pairs of hooks that is infective for the intermediate host, known as the hexacanth embryo or oncosphere. Transmission, ingestion of the larval stage called the sister circus, Larvae consisting of a fluid-filled sac containing invaginated scolex, cistercoid, or plerocercoid larvae found in raw meat or fish of insects harboring the larvae stage. So here are the eggs of the important players here and their relative size. So a lot of these kind of look alike, but it's the size that helps you differentiate them. And a lot of times when you see a familiar shape under the microscope, you then go and measure the egg microscopically, and then you look at it compared to a reference manual. And the reference manual has all the different types of eggs and the relative sizes that they should be, and you compare it that way to differentiate. Identifying features are listed here, so you can make note of these identifying features, and maybe these will help you when you're learning all of the egg shapes so that you can easily identify these on an exam. The diagnosis of tapeworm infection is usually made by finding eggs in feces, although proglottids can be used if they're passed intact. Intestinal tapeworm infections are usually treated with praziquantel. Alternatively, they can be treated with niclosamide, albendazole, or nitazoxanide. Diphyllobothrium latum. So this is the fish tapeworm. Adults can be 10 meters long. You ingest the larvae in pickled or raw freshwater fish, and this is found in the Great Lakes areas. Humans are usually only harbor a single worm. It attaches to the jejunum of the small intestine. Clinical infection is usually asymptomatic, but sometimes can cause nausea, vomiting, intestinal irritation, vitamin B12 deficiency leading to megaloblastic anemia. And that's because when the tapeworm attaches to your intestine, its goal is to absorb your nutrients. So when you bring in food from your diet, you're going to digest it in predominantly like the duodenum and beginning part of the jejunum. And then your small intestine is going to try to absorb nutrients from your food. Well, this guy's also going to be in there trying to absorb your nutrients. And it selectively reabsorbs or absorbs a lot of vitamin B12. So the life cycles are important. You should be able to say the general life cycle of all of the ones that are depicted in this lecture series. So this week's, next week's, in general, you should know the life cycles. So the eggs are in the water. The choroncidium is released, and then crustaceans are going to consume these. Then the fish is going to eat the crustacean, and then the person's going to eat the fish. And they say raw fish because if you cook it, these things die. Then the larvae is released and it matures in the intestine, and then you have the adults in the intestine of the human. So the adult worm then grows. Eventually, the lower end will start to release the eggs in the feces. And then depending on what type of water source, sometimes these eggs get into the water source. The diagnostic structures that can be found in fecal samples, the scolex, proglottid, or egg. Egg morphology, it says the size, small knob-like protuberance at the end opposite of the operculum. So just want to kind of skip ahead to this picture. So the distinct operculum, let's see that here, and then there's the abopercular knob. So you can see a little knob sticking out of the bottom part of the egg. So this is kind of like an opening here where the worm will emerge from, or the larvae. So you can see that at the top and this at the bottom. The knob may not be seen on all eggs, so the size and lack of shoulders must be used to distinguish the egg from Paragonymous westermanii, or westermanii. Proglottid morphology is wider than it is long, characteristic rosette shape or coiled uterus. So here's another picture of D. latum. And then again, here's some pictures of it blown up. And then here's some pictures of eggs, scolex, and the proglottids. Scolex morphology, 2 to 3 millimeters long. Elongated, has 2 suckling grooves, one located on the dorsal surface and the other on the ventral surface, and there's no hooks. And here's a picture of that scolex. So Tinea. Tinea species that infect humans would be Tinea saginata, which is the beef tapeworm that's found primarily in beef-eating countries in the world, and Tinea solium, the pork tapeworm, which is found in areas of the world with high consumption of pork, such as Latin America. Both organisms attach to the intestinal mucosa of the small intestine. So here's the life cycle where we start with eggs in the feces. So eggs in the feces, Tinea solium only, the human would ingest the eggs, then it's released into the tissues, and these will form cysts inside of the tissues. In the eye, the brain, the muscles, or the bone. If they do form cysts in the brain, that is an extremely bad thing. Obviously, we don't want cysts formed in the brain, and that can cause someone to die. So many cysts can then implant in the brain and can implant other places in the body, causing severe cognitive deficits and even death. If we talk about the eggs in the feces, ingestion of hosts by an intermediate hosts, then those would be released into the intermediate hosts, and then those cysts would then either go into the cow or they would go into the pig. So if they're T. saginata, they would be in the cow. T. solium, they would be in the pig. Now, when we consume the cow or the pig, typically we would cook the meat before we consume it, but if it's undercooked, even smoking the meat does not kill these. So if it's undercooked, then we would bring it into our digestive tract, then it would attach to the intestine, and it would cause the tapeworms to grow in our intestine. The adult tapeworm would then release eggs, and those eggs would then be passed in our feces. So a couple different life cycles here. In this life cycle, we are consuming the animal meat that's infected with the cysts of this organism, and that's how we get the tapeworm in our intestine. In this cycle over here, we're actually ingesting the eggs, and if we ingest the eggs, they can encyst in our muscle tissue or in our brain or eyes or bone, and that can cause some very detrimental effects, including death. The length of the adult worms are a little bit different. T. saginata is a little bit bigger because that's the cow one. So T. solium is the pig one, a little bit smaller. Symptoms: infection with the adult tapeworm of either species usually causes a few clinical symptoms, although vague: abdominal pain, indigestion, loss of appetite. The major complication of infection with T. solium is cystericosis, in which the infected individual becomes the intermediate host and then harbors larvae in the tissues. Eggs are indistinguishable. The proglottids, though, are a little bit different, and I'll show you some pictures in a little bit of how to differentiate T. solium and T. saginata by their proglottids and scolices. So the primary method of diagnosis of any Tinea species would be the eggs in the stool. Again, eggs between the two species are indistinguishable. If you get the adult worm, then you can distinguish them based on their scolex or proglottids. So this is the beef tapeworm, and you can see the proglottid structure. The eggs are indistinguishable. And here is the proglottid structure of the pork tapeworm. So if we kind of go back and forth, you can see that there's many more of these uterine lines as opposed to here. There's fewer and they're wider for the pork tapeworm. And then if we look at the scolex. So here's the scolex of the pork one versus, the beef one versus the pork one. And then here you can see the differences. You can see the lateral uterine branches of the proglottids. In the beef one, they're a lot thinner. In the pork one, they're a lot thicker, and there's fewer of them. Hymenolepis. So Hymenolepis, the dwarf tapeworm, is Hymenolepis nana. It's found worldwide. It's a common tapeworm in children. Hymenolepis diminuta is the rat tapeworm, and this one is seen a little less frequently. Infections are usually asymptomatic. Large numbers of worms may cause abdominal pain, diarrhea, irritability, and headache. H. nana is easily transmitted amongst children, and an intermediate host is not required. The life cycle: direct fecal oral egg transmission of the egg, development of the cystrocoid in the intestinal tissue of the host, re-entry into the lumen for development into an adult, and this autoinfective cycle is most common, although insect vectors may serve as intermediate hosts in alternative morphology of the life cycle. So here's a diagram. You've got eggs in the feces. The person consumes the eggs. It migrates to the bowel, and then it forms cysts and eventually grows into a large worm. And then from here, they're going to, those cysts migrate into the intestine and grow to adult worms. In this instance, the eggs may be carried by certain types of insects. The embryo goes inside of the insect, and suddenly it penetrates into the human when the human accidentally ingests the insect. The adult H. nana morphology: approximately 40 millimeters long, a small scolex with 4 suckers, and a rostulum with spines. Primary diagnosis method would be eggs in stool. The egg morphology is listed here. And this is what the egg of H. nana looks like. And here's another picture of the egg and the actual worm. Here it's encysted in some tissue. Hymenolepis diminuta, 20 to 60 centimeters long. Morphology of the egg: it's a little bit larger, gray or straw colored, oval. And this is what that looks like. Dipylidium caninum: humans serve as accidental hosts for Dipylidium caninum, the dog tapeworm. Children are usually infected by ingesting fleas containing the larval stage. So for this one, fleas are on the animals. The fleas then jump off the animals. Because kids are all over their pets, if your pets have fleas, the fleas can accidentally be ingested or jump into the mouths of the kids, and then they can release the larval stage, and the larval stage will then grow into a tapeworm inside of the child. Resulting infections generally do not cause any symptoms. There's proglotid and egg morphology is listed. You will not need to identify this on an exam for this class, but this is what they would look like. Tissue infections with cestodes. So Cysticoccus, sparagonosis, and hydatid cysts diseases are the major diseases caused by the tissue stage of a tapeworm. They originate when a human accidentally becomes the intermediate host for the parasite. Cysticoccus results when a human ingests the infective eggs of T. solium, the pork tapeworm, thus becoming an intermediate host. Geographic distribution: endemic in rural Latin America, Asia, and Africa, reemerging as a zoonosis in the United States as a result of the immigration of people from endemic areas. Contributing factors for infection: poor hygiene, poor sanitary habits, residence in rural areas, or hog farming. Life cycle notes: the egg is ingested, hexacanth embryo is released into the intestine and penetrates the intestinal wall, entering circulation, and then it develops into a cysticosis in any tissue or organ. The larvae can live up to 7 years and elicits a host tissue reaction, resulting in the production of a fibrous capsule. Once the organism dies, it releases larval antigens. There is an intense host inflammatory reaction that leads to tissue damage in the area. Eventual calcification of the Cysticercus will occur. The most commonly infected sites are the striated muscle, eye, and brain. Light infections usually cause no clinical symptoms, but the symptoms depend on the organ affected. The size, number of cysticerci present. In striated muscle, pain, weakness, cramps in the eye. It can cause retinal detachment, eye pain, flashes of light, blurred vision. In the brain, this is the most serious manifestation, so neurocysticercosis, the causative agent for up to 30% of epilepsy cases seen in patients in countries where the disease is endemic. Other possible manifestations, headaches, symptoms resembling those seen in meningitis or brain tumor, convulsions, or a variety of motor and sensory problems. The appearance and diagnosis are listed here. So for diagnosis, a radiography, we can do an eye exam, imaging like CT or MRI, histological tissue staining, serological tests. This is a diagram that will help you differentiate the proglottids and scolices of the main species that you need to be able to differentiate. So plan on studying these images. You will see these images again in future questions. And then this is a picture differentiating the major morphologies of the different types of tapeworm eggs. Again, you're going to want to study these pictures. And then here, these are pictures of microscope specimens of organisms that I actually identified from samples. So you can take a look at what I actually saw under the microscope.